Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Sawsan Youssef. She's the Chief Scientific Officer at Distributed Bio, and we're going to talk about the work that she does there. So, Sawsan, thanks for coming. How are you doing today?
Thanks for having me. I'm doing great.
Oh, good. So tell me about Distributed Bio. What is the company about?
So Distributed Bio is a very unique small biotech company that is based on immuno-engineering, basically engineering new technologies for developing new therapeutics in the field, in any medicinal field, using immunology as the platform.
Okay.
And generating different platforms to develop antibody therapeutics for from inflammation to cancer to viral vaccines. And the uniqueness about Distributed Bio is we are self-funded, so we don't rely on venture capitalists. We support ourselves. So part of using these platforms as a way of generating as a CRO services to generate therapeutics, antibody therapeutics, using our unique platforms and technologies for other companies while we're developing our own internal portfolio. So we have in our internal portfolio, we have two types of therapeutics. One's relying on antibody generation, and one is vaccine. So I'm pretty sure you guys also heard about us through the COVID-19 pandemic. But for that, we are developing a therapeutic, which is an antibody therapeutic and not vaccine. But we also have these technologies for like flu vaccine and HIV vaccine that we are developing internally.
Okay, with that, let's talk about antibody therapy and vaccines. So how would antibody therapy work, for any disease or condition?
So antibodies are a very in therapeutics, there is two areas. There is the small molecules and there is the large molecules with antibodies. So antibodies actually are really good therapeutics in a way. They are very tolerable in the human body, and their effects are directed against the target that you are aiming for. For example, if you have a molecule that is important for that initiates cancer, for example, if you generate an antibody, like for example, like HER2, you generate an antibody against that specific molecule, so it's targets specifically that molecule, and the side effects will be very minimal versus if you give those patients like a small molecule that affects many pathways. So there's maybe the effect will be as strong, but it will have a lot of outside scope side effects because affect a lot of other pathway in parallel. But sometimes you have no choice. You can find a molecule for this type of disease or not. So to target as an antibody therapeutic, so you have to use a small molecule therapeutic. So it depends on what target you wanna aim for vaccine.
Quick question here. So how do antibodies work? Do they bind to a target and then they attract immune cells to engulf it, or do they choose to do some other action as well?
Yeah. So basically, what the antibody does, you can engineer it, and we are very good in that here as well. You engineer it based on what you want to aim to get out of the therapeutics. So, for example, if you want to block something, one molecule on the surface of cells, what you generate is an antibody that will block this molecule and this protein and stop the signaling downstream. So this once the antibody, and usually the antibodies are made in such high affinity that you will prevent from other normal protein that bind this target in the body to bind, because it binds so strong. That's what mean high affinity. It binds so strong. You make it so strong that it will not let it, until this molecule disappear, you won't let it leave. So now these antibodies can be engineered in many ways in their profile to, if you want to induce like an immune response, so you put this antibody in such a structure that will recruit these in the other side, where there is the side that bind the target, and there is the other side that can induce the response that you want. So if you want to bring T cells and B cells and macrophages to kill this let's say tumor cell, then you will engineer that antibody in a way that in the other side, when it's bind the protein that we want to target, it will start this cascade of inflammation and immune response to generate this response. But if you just want it, like for example, for COVID-19, for example, because there is some thought that antibodies might cause extra inflammatory response. So you want to generate an antibody that just bind, for example, the virus and prevent it from infecting cells. So then you design other type of antibodies against other viruses. So you design it in a way that the antibody will bind the target and just block it from what it caused, like for example, in virus to infect cells. So it depends what do you want to generate.
Right. Yeah. I've heard that sometimes antibodies can be binding. They'll bind to a target, but sometimes that'll make the target even more problematic. And then there's some that are neutralizing. So they'll bind in such a way that they will stop the bacteria or the virus or whatever it is from causing harm. So I guess there's a difference, right?
Yeah, so you generate so the side that the antibody has like a Y shape. So in the top of the Y's, like the Y shape, you have the binding of the target itself. So in that side, you won't change anything. But the stem of the Y, you can mutate, or you can make the antibody. Of course, it will be very similar to human antibodies, because we know exactly how human antibodies look alike, and there is multiple. There is 1, 2, 3, 4. There is different types, and you design your therapeutics similar to what the human body generate, but you can make some kind of adjustment in that stem of the antibody to either cause, in case like for cancer, you want to bring a lot of immune cells to kill the cancer cells. So in there, you generate that antibody that will look exactly like the antibodies we generate in general in our bodies, but you modify it in a way that it will cause the killing, for example. Or if you want a neutralization, you generate an antibody in a form that it will give you the neutralization only. So it all depends.
Yes, I guess you got to pick targets on the surface at certain spots and bind to them to do it the right way. So what are some of the strategies? Do you like for a virus, I guess you'd want to bind to the spike protein and not just to the to the capsid. Like what are some of the strategies to make an antibody work better in terms of where it binds or how it binds?
So we here in distributed bio, we develop technologies that we can also generate multiple First of all, we do our discovery process where we find those potential antibodies, and we, based on functional assays that we test after we have this profile of antibodies, we choose the most probable the most probable part of this antibody to give us the optimal binding property in the optimal binding sites, because antibody can bind different sites. So you set up, after your discovery part, you set up several assays that you test, and you pick your most probable candidates. And then if you want them to bind very, very strong, we have different also next step technologies where we kind of mutate sites in those antibodies and make them higher affinity or binding different epitopes or same two epitopes at the same time, like parts of whatever protein we want to generate against. And then after you finish that part of discovery, then you go ahead and you say, do I want to make the antibody just bind and neutralize, or do I want to make the antibody with some effector functions, meaning like causing the cells to die or not? And there, where you plug your small parts of the discovery into the big backbone, and then you have your final drug therapeutics, and you go through intensive assays and intensive detection assays to decide, okay, I think I chose this antibody because it binds this part of the target, and I would like to have this function or prevent this function. And that's how you get to the final product that you move into the clinic later on.
To make sure that the antibodies are as close as possible to natural antibodies in terms of them also having the Y shape, being similar size and all that stuff, or can you make them very divergent?
No, you want the antibodies to be as close as to the human. It's called germline, meaning the human natural properties of antibodies, because you don't want these antibodies, once you treat the patients with, to start the body, then if they are really different, the body then will be starting to see it as an anti-self, and then it start to generate another response against it and then reject the antibody. And that's very common sometimes, but in antibody therapeutics, this side effect is very low, because the engineering, and here at Distributed Bio, we are very good at it, because we generated our library to eliminate those immunogenicity, what is called immunogenicity parts from the antibodies. So the final products are very stable and very humanized as we can get them. So the probability that, again, it's a person to person, but the probability to get this side effects is very minimal, and that's where we are very good at.
Are you saying that if you make the antibodies very unusual or different or alien, the body mounts a response, and you may get an overreaction from the body, and the person may get sick and die, let's say?
No, they won't get sick and die, but they will have to stop the therapy in order to, they will have some side effects like rash or different. I mean, each person will have, and each therapeutics will have different side effects, but they have to stop the therapy for them. So in general, in companies and knowing all these immuno-engineering and the immunology of antibody therapeutics helps us primarily to generate antibodies that as human as possible, to avoid all. And that's where we were excelled, because our founder and CEO Jacob Glanville had generated these antibody libraries and took in consideration all these problems and of immunogenicity, of foreign or only. There are also antibody alleles. So like they're antibodies that will work only with one type of population versus the other. So we eliminated all of that, and we made our antibody libraries for discovery as human as possible and as general as possible. So it will lead us to the optimal. So we built ourselves for success to lead to the optimal discovery platform, and we developed these technologies to be the discovery part will go very, very fast in like a month or less than a month to generate those preliminary pools of our antibodies to go quickly into screening and generate the final product.
What's an example of a proper immune response, and what's an example of an improper one? You know, if you make the antibody just right, what will happen? Maybe a few details, and if you don't, what can happen?
So it depends what you want from the antibody, and I will give you three examples. So if you want an antibody against a target that, let's say, that's the easiest way on cancer cells, like let's give an example like HER2 for breast cancer, or let's give an example for CD19 for leukemia. What you want from the antibody is to go bind that bad target, and you want to bring as much as immune cells into that area where they cancer to kill the target, to generate killing of the tumor cells. So you choose the antibody that binds this target the strongest and the more specific, because sometimes our body generate proteins that are very similar between tissues. They're different but similar. So you want to have your antibody specific only for that target, and you enhance that effect with having a Y shape that will once this target binds. In the other side, it will bind to other immune cells they will start the inflammation, like bind to macrophages. Macrophages go call T cells and B cells, and then you start getting a huge immune response in the side of the tumor, and you kill the tumor. There is other antibodies that you want them to bind immune cells and go. That's what's called immuno-oncology. And go bring those immune cells so what happens in general, let's say when you have and I'm talking in the cancer area right now, but we can change it into inflammation as you flip the backbone. And I will explain in a second. So if you want to, like, let's say immune cells go in general to kill the tumor cells without any antibody therapeutics. So there, what you do is those cells started to get activated and start to kill the tumor, but they get what we call them exhausted. They're not good enough to finish the job because they get exhausted, because they start expressing some proteins that the tumor knows those proteins, and they tell them, okay, stop, don't continue. So in that case, we generate an like all the PD-1 and CTLA-4 that were all in the immuno-oncology field that came out a few years ago and changed the whole concept of many cancers like lung cancers and bladder cancers and gastric cancer, where basically you go and this antibody will bind targets that on your own immune cells. But because you don't want your immune cells to die, because what happens here is just the antibody will bind those molecules that they call it checkpoint, where they stop the immune cells from acting. So those antibodies, you don't want to have you just want them to bind the cells. So once they bind like the T cells, then the T cells started, I need to start getting activated and multiply again and again and again. And then you increase the immune cells in the tumor and you attack the tumor. So here you basically blocked your exhausted immune cells from being exhausted, and you facilitate them to kill the tumor themselves. And that's basically an antibody that you generated to just bind the target and tell the target stop. And there is other antibodies that you actually want to increase the response. So you basically, it binds the target, but it binds the target in agonistic way, meaning that it will bind the target and then tell the target, okay, I want you to start getting activated again, and then get the immune cells or get any other cells that you're attacking to respond to this target. It will see it like as a ligand, and it will start basically get activated again. So it's all about the engineering of what you want your antibody purposed for, and that's how you just manage from binding the target and making it to final human and make it to the assay that you want to generate and get. And
okay, I got it. When the Y arms, I guess, of the antibody bind, does it change the tail? Does it change the structure of the tail to make it active or inactive or different? So you're able to design one that way?
Yeah, so once it doesn't change it, it just depends on what tail is attached to then, but it has to bind the target first, and then the tail will know what to do. Either will bind other cells or just block. But there is some conformational change between, once the binding happens, there is a little bit of conformational change there.
So what happens, I've heard with some viruses, if there's only binding antibodies, it actually enhances the ability of the virus or allows it to find another way to enter cells, and then it's even worse than before. It's like an antibody modulated, entry. Can you talk about that briefly?
What happens there in that case, if new viruses or so there is two ways. If there are some viruses that are highly mutated, that they, like, for example, flu virus, every year we have to get a new vaccine because they change, they rapidly mutate. So if you generate one antibody against them, or HIV viruses as well, they are highly mutated. So if you generate an antibody against it, the replication of the virus it change very, very quickly, that they might change their own proteins in a way that the antibody will not be able to bind. Or in that case, you want to generate a vaccine that kind of give you a large or a spectrum of vaccine that will recognize a different potential mutant that will give you a spectrum of protection. There are some viruses that they don't change very much in that protein, but what happens is that they do have multiple proteins that can initiate the entry of the virus to the cells. So if, like, for example, in the coronavirus, the new COVID-19 initiating disease virus, there is one site that we know, but because it's new, we don't know how many other proteins will also facilitate from the envelope of the virus, facilitate the entry of the virus. So in that case, it just more research and more science need to be done to understand that. So for that case, for viruses that are known that have few entries, you either again generate a vaccine, or you generate combination of antibodies that block several protein that helps the virus infiltrate the cells, and you basically block all the possible entries, and you treat the patients, if you're going into antibody therapeutic, with the cocktail, like they did for Ebola, for example. They give a patient a cocktail of 3 antibodies, the Regeneron treatment, and they if the patients come really fast to the clinic, they 94% of them survive, which was a few years ago a really bad pandemic. So we need to understand the target very well in order for us to be able to design the optimal therapeutics for it.
Yeah, makes sense. Okay. Anytime I ask you anything, if it's proprietary, obviously, don't answer. So, no worries there. What's the current work that you're doing that you think is showing some promise, moving forward? Are you very close, you think, to a breakthrough either with COVID or with other diseases, or just in general, making these antibody libraries, for some reason or somehow more effective than they currently are?
Yeah, so I can talk in general. I mean, I'm pretty sure you guys heard about our COVID programs because it's in the news. So I mean, we are working to generate an antibody therapeutics for it. We think we have interesting data that we will follow up on in the future with press releases, etc. And we are moving aggressively to going into first in human, hopefully by end of August, September timeframes. For there is a few internal projects that I can't talk about very much, but I can just briefly touch base. Using our technologies and amazing libraries, we have a very interesting target that might be relevant for hard autoimmune diseases like lupus and arthritis, and that if our antibody therapeutics seems to be very promising, but that will be we're still in the development, because the speed of the COVID is already going really, really fast, and we are in preparing for filing in the next few months for IND filing and going into human. For the other inflammatory diseases like lupus and arthritis, it might take a little bit longer, just because both COVID prioritized, and then it's a longer process. Usually antibody therapeutic development take in normal 18 months from discovery to going into first in human. So for that one, we are in finalizing the discovery and finalizing the final candidate, and hopefully in a year or so we will be going into filing. There is another project that I'm working on specifically, and that's, I won't talk about it too much because it's still proprietary, but I'm working on finding a way that where immuno-oncology therapeutics are really helpful, but not helpful for all diseases, and we think we found a pathway to overcome that and use those immunotherapeutics in cancer patients that don't respond to them by giving them the supplementary technology that we are developing. And so far, so good. But that probably also will be highlight in the next year or so, because we are generating the data to prove it. So we prove it in first step. Now we're proving it second step, and so that project is really important for me, because in my previous company, I worked on immuno-oncology target. I generated a PD-1 antibody, and I noticed how and I was involved in the clinical trial, and I saw certain patients respond and certain don't. So I always think about the patient who don't respond, and I thought about it many times, but I didn't get to the idea until I moved here and I started thinking about it and studied about it, and it's just we'll see how it goes. So
I have a quick question about attacking tumors, by attaching antibodies, since tumors are heterogeneous, and I'm sure as the more that a tumor goes along and metastasizes, the more heterogeneous it is. Mm-hmm. If you were to create a way to attach antibodies to it, I would think that you would need to have a cocktail of different antibodies, because a given antibody would probably only attach to a given cell type, and ones that are differentiated away may need different antibodies. So, you might only kill some of the cells of a tumor, not other ones, and the other clonal lineages would keep going.
That is totally correct. And a lot of companies has done these studies, but again, the tumor cells are so smart, they escape, and those combination therapy right now are successful, but not as expected, because the tumor has many faces and they're still learning about it. So right now, like the best combination is giving a checkpoint inhibitor with a radiation or with a chemotherapy. But still, like the mixture of the antibodies, in my previous company and many big pharma, they have tried this, but it appears that if the tumor responds to an antibody, it will respond to the cocktail. You will maybe increase in the same population the percentage of responders, which is great, but you won't completely bring a different population that don't respond unless you try with a different therapeutics, meaning like chemotherapy plus antibody or radiation plus antibody. But in my thinking, that if this technology works, you just need to give that, and then you keep following up with these antibodies, and you can escape other harsher treatments for the patients. But this is still going on, very, very beginning, and hopefully we'll see a progress very soon, and we'll decide how to move aggressively about it.
Right.
Because still, like with all the pandemics, still a lot of patients die from cancer, and not all cancer patients have the potential for all these therapeutics that are developed, because they have something resistant in their tumors that those therapeutics are still not there yet.
You mind if I asked you just for a few minutes? I didn't ask you much about vaccines. We talked about attaching antibodies and causing immune responses properly, but with vaccines, can antibodies be used to bind and turn an invader into can it help the body create a memory of the invader?
Yeah.
That it becomes its own vaccine? Or like, how do vaccines work in regards to your work?
So the vaccine, what you do the vaccine for is to basically teach your immune system to develop those antibodies for this certain virus or certain bacteria or whatever you want to, or even there is vaccine for cancer. To develop, you teach your immune system to generate those antibodies and develop them and go attack the bad invader, whether it's a tumor cell or a virus or a bacteria or something, some parasite or whatever. The thing with the vaccine is it's built on making your body have a memory, which means sometimes you need maybe multiple doses to give the antibody, so you can educate your cells that have never seen this foreign target to have an immune response and treat this target. And that would take at least a month or 2 months until you build your immune response in the case of viral or bacterial infection. But in case of a tumor cell, it might not be sufficient to eliminate, because tumor become sometimes like you said, metastatic or very aggressive. But it helps on top of the other therapeutics. But when you have like a sick person, and vaccine, you have to give them ahead of time, like we all go to get the shot before the flu season, because we want to either make a new cells that will respond to or we want to wake up the old cells that we had sitting, and because they didn't see that virus or that bacterial or target, and to activate it again. So it does what the antibody therapeutic does, but it takes a longer time. But when you have like a sick person, like for example in this pandemic, you want to do the vaccine part outside in a big facilities that will generate these antibodies for human treatment and use. So you basically do that part of education outside by the technology and by the engineering outside of the body, and you give the body that drug to block the disease quickly and not wait on the vaccine to work, because some of these patients are really, really sick and they need some kind of therapy to block it on time. So that's basically the difference between vaccine or therapeutics, antibody therapeutics, that in the antibody therapeutics, you try to block the disease or the infection agent exactly when you inject, or in a day or less than a day, or 48 hours. But for vaccine, you basically educate your body that when the next wave comes, our body will be ready, and it by itself will fight this pathogen.
Okay, well, very good. Well, Sawsan, thanks for coming. It's very, very interesting. And how can people find out more about your work at Distributed Bio?
So they can log into our website, distributedbio.com, or we also are having a spin-out called Cinevax, where we're going to move all our therapeutics into the spin-out. So it's you can also look at us at cinevax.com, either on the website, or we have links in LinkedIn or Facebook or Instagram. So you can go to cinevax.com or distributedbio.com, and Twitter and Facebook, Instagram, LinkedIn, and find more information about us and about our company and founders and us too.
Very good.
And the research we do.
Well, Sawsan, thank you for coming. I appreciate it.
Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.